One. Hello and welcome to the RX Hope Table Talks. This is session 5 on gene therapy techniques, AAV and CRISPRa for SCN2A. My name is Jenny Burke. I'm on the Family's SCN2A Foundation board. We're very excited to be bringing you this virtual educational experience to our global community. During these sessions, families and caregivers of the patients with SCN2A related disorders will be referred to as the experts, and the presenters will be the professionals. The goal of the virtual table talks is to allow experts unprecedented access to professional researchers and clinicians working diligently to find a cure for our children. This is meant to be a casual environment, and your questions and comments are encouraged as we're all here to learn from one another. To keep things running smoothly, please keep your audio on mute and use the reaction buttons to raise your hand if you would like to talk. Alternatively, you can send questions through the chat. If you haven't already, please introduce yourself and your SCN2A child in the chat, perhaps their name, age, how SCN2A presents, and where you live. And I now have the pleasure to hand this over to Stefan Sanders from the University of California, San Francisco, who will be talking about gene therapy techniques. Take it away, Stefan.
Thanks for the lovely introduction. Let me share this. There we go. And let's go on full screen. Great. There we go. I trust everyone can see that. Fantastic. There we go. So my name's Stefan. I trained as a pediatrician in the United Kingdom, as you may be able to guess from the accent. I then went to Yale to do a PhD in genetics. I now work as an associate professor at UCSF, and my lab specializes in bioinformatics, particularly in trying to understand the cause of autism, what that cause does to the brain, and recently, the last few years, what we can do to try and improve it. And one of the foremost examples, in fact, the foremost example which we study is SCN2A, partly because it's the gene which comes highest in our evidence for autism association, but also because it's a gene where we kind of know what it does, which is a really good starting point for biology. So in fact, the picture on the right there is the SCN2A channel. So it makes a protein called NAV1.2. This is the protein and what it looks like. And that little hole in the middle there, that's where the sodium goes through. So I'd just like to be sort of frank and give disclosures here. So I do receive research funding from BioMarin and collaborate with them. I think for this group, that is probably good news, because working with pharmaceutical groups is part and parcel of trying to bring some of these techniques into translational activities. Given the title of this, I wanted to start off with a message of hope. In fact, a message given by someone else, someone who really is in a position to comment on this, and that's Monica Cohen-Rads, who's the Chief Executive Officer at the Rett Syndrome Research Trust. And she recently put out a web blog saying this idea there's a reason to celebrate that children with severe neurological disorder improve after gene replacement. To me, this is a big thing. And the research she's talking about is in 2 different disorders. One of them is a metabolic disorder. I'm not going to try and pronounce it, but you can see it written there. And that's a gene replacement therapy. And the second one is an Angelman syndrome, and that's using an antisense oligonucleotide, and we're going to come to those later. In both these disorders, they cause severe developmental delay. In both these disorders, children who'd had the disorder for multiple years were given a treatment and showed some signs of improvement. And this is one of the big questions we don't know. When can we reverse the symptoms? And we have no examples. To my knowledge, these are the first 2 examples of someone trying to genetically treat one of these conditions. And in both situations, improvement was seen. And so I think this is a real message of hope. There are some disorders where you can get improvement in these symptoms later in life. I truly hope that in the not too distant future, we can add another gene to this list, and that gene, of course, being SCN2A. So I'm just going to start off with a little bit of biology. I expect to most people in the group, you've heard this, and it's probably a bit of revision. But I think in terms of making sure we're all on the same page, I think it's very, very important revision. So here's what the channel does. I'm simplifying it quite a lot, but basically SCN2A makes a protein or a channel called NAV1.2. That channel sits in the membrane of a neuron. And it has one job, and that job is to sense changes in electricity in the electrical environment, and in response to that, open and allow sodium in. And that influx of sodium creates an action potential, which is how nerves fire and how nerves communicate. So it's a really, really fundamental role. But the key thing here is that it's all about the amount of sodium that comes in. Some individuals have a mutation in their DNA. And this is the most common pattern we see in SCN2A is a de novo mutation, a change in the DNA which is not seen in the parents, which is new to the child. That means that's why we call it de novo. And that mutation sometimes changes one of the building blocks of this channel, AAV 1.2, and sometimes it just stops the whole thing from being made. But this is kind of approximately to scale. It is just a tiny, tiny change, but sometimes it hits somewhere critical and has a big effect. And we see broadly two major impacts of having one of these mutations. On the left, we see infantile epileptic encephalopathy. So this is really characterized by early onset seizures in the first year of life, sometimes on the first day of life, sometimes before birth. And there are anti-epileptics which can modify that, but rarely completely control it. And that's then also associated with learning disability, for which there are no real effective therapies. I think we think that that is probably somewhat secondary to the seizures, or at least part and parcel of the same thing. And then the second major group is autism and developmental delay. And these children tend to present first with social communication impairment or a learning disability or failure to meet developmental milestones at the right time. And at the moment, there are no effective therapies for that. And then some individuals go on to develop late onset seizures, for which sometimes anti-epileptics help a bit, but again, rarely completely control it. And through work done really with Kevin Bender at UCSF in collaboration with myself, and then also independently done by a group in Europe with Rike and Marcus Wolf, we've seen this model emerge that if this is the normal situation where the channel is meant to let this amount of sodium through, you can have a gain of function mutation, which leads to too much sodium coming through, or sometimes sodium coming through at the wrong times. And that leads to the epileptic encephalopathy. And on the other side, in the face of autism and developmental delay, we have a loss of function mutation, which leads to too little sodium coming through. And this leads us to a relatively simple model of what we're trying to achieve in our therapy. On the gain of function side of things, ideally, we would have less SCN2A, trying to reduce the influx of sodium. In contrast, for the loss of function mutations, we're trying to have more SCN2A to try and increase the influx of sodium. I would note this is a slight oversimplification. We're dealing with, of course, a very, very complicated structure of the developing human brain, and that this is a little bit simplistic because you can have maybe some different types of gain of function. You can also have a mixture of loss of function and gain of function. But if you had to do one thing, if you could change one dimension of the disorder, this would probably be it, an increase in the loss of function and a decrease in the gain of function. So there are three broad ways of achieving this. The first one on the left is small molecules. And this is kind of the medicine which we're all familiar with. This is taking a pill, or sometimes maybe having a little injection into the skin, but most often small molecules which are ingested orally in the form of the pill. And the idea here would be that you modify the function of the protein, the channel NAV 1.2, either increasing it or decreasing it. This is something which is being actively pursued. It is made very difficult by the fact that NAV 1.2, which is the one made by SCN2A, is very, very, very similar to a gene called SCN5A, which is critical to the heart. And so the risk is if that small molecule sort of touches SCN5A, it could lead to a heart problem, which of course is going to be a very, very, very challenging problem. And then similarly, SCN2A is very similar to SCN1A, SCN3A, SCN8A, all of which have critical functions in the brain. And so for that small molecule to be successful, it needs to be very, very, very specific to SCN2A. And that's something people are working on, but it is a very challenging problem. On the far right, we have AAV delivered genetic therapy. So this is probably, when you hear about gene therapy in the news, this is usually what you're hearing about. And this is the side of things which Serena is going to talk about in the next talk. And doing this, you can deliver different things. Probably the simplest thing you can do is just send the missing copy of the gene. Unfortunately, SCN2A is too big for that. So that doesn't really work in this disorder, but it was one of those first examples I gave. Serena is going to talk about CRISPR or modifications of CRISPR, which are ways of modifying this in a more nuanced way. And the dream here is that you have a single shot, and that's it. That single shot modifies your DNA, hopefully forever, or at least for a very, very long time, for years, leading to a long-term improvement. But that's a permanent change. And in the middle, we have antisense oligonucleotides. I'm going to go into what those are on the next slide, but this is sort of somewhere between the two. It's sort of gene therapy, like it's very, very specific to one place in the genome, but it's reversible. It's a non-permanent change. And so it's kind of gene therapy light, or the way I see it, a really nice stepping stone before doing some of the more permanent CRISPR changes. And with this, in theory, you can increase or decrease the expression of a single gene. And what this would look like as a therapy probably would be an injection into the spine every 3 months or so, a little bit like having an epidural. So that's obviously far from ideal. It's a lot more involved than, say, taking a pill 2 or 3 times a day. However, it does last for 3 months, and it gets into the sort of the brain region, which is why into the spine is the place. People are working on making these so they can be delivered in different mechanisms, but right now, that's what it would look like. So I'm going to talk now about what an antisense oligonucleotide is. So here's a picture of some DNA on the left, the beautiful double helix. So DNA gets transcribed into RNA, and then that RNA eventually will go on to make a protein. So the RNA is different from DNA in that it's a single strand compared with the DNA, which is a double strand. Now, an ASO is very simply the opposite of that RNA. So if I show the last picture, you can see these two fit together because it's the opposite. But with the backbone here has been chemically modified. And that chemical modification does 2 things. It firstly makes it really hard for cells to break this down, and that's why it lasts for so long. And secondly, it means that when it binds to RNA, it really sticks. And that's why they can have such a dramatic effect. And so with them, you can accomplish a number of different things. Depending which bit of the RNA you target, you can perform many, many different modifications. But I'm going to try and keep it at a high level here. And broadly, there are some approaches which decrease gene levels. An example there would be the ASO, which has been used in Angelman, where it's targeting a gene called UB3A antisense and decreases that. The other thing you can do is increase gene levels. And this is a very, very new idea. The first hint of this came 3 years ago. And there's an example which has been done in Dravet syndrome, which is our colleagues in SCN2A. So let's think about what this would mean in the world of SCN2A research. So if we want to decrease the amount of a gene, that is relatively simple to achieve. Putting it in the simplest terms, it's quite easy to break things. It's much harder to make them. And if you're trying to decrease the amount of expression of a gene, that's in the breaking realm, which generally is easier. And so the way that would work, on the left, we have a child with a gain-of-function mutation. They've got one normal copy of SCN2A with a normal amount of sodium coming in, and one gain-of-function copy of SCN2A with too much or it coming in at the wrong time. The ASO would decrease SCN2A, leading to this pattern on the right where both copies have been decreased. Now, this sort of captures, I think, the problem with this, or the challenge with this, is that you're not reverting it to the completely normal state. You're just damping down the abnormal state. So there's probably a ceiling on sort of how much effect this can have. And then secondly, we know that if you have too little SCN2A, that leads to the loss of function. And so potentially there's going to be a very, very narrow dosage window where this might work. But as a concept, I think this is something which is well worth exploring and is something which is being actively pursued by Dr. Steve Petru and his team based in Australia, and already is at the stage of being in animal models with examples there. Now, there's one other challenge with ASOs, and that is that if you design it in a mouse, it works in a mouse. It's not like other medicines where you can sort of change between species, because the bit you're targeting, the bit of DNA or RNA, often differs by species. And so certainly in Angelman, we've seen that while there was a lot of work done in the mouse, there was then that whole work had to be largely redone in humans and human cells. And so the other side of things, trying to increase the amount of expression, is much harder. You need to be lucky. You need to be able to find a sort of genetic trick to be able to increase the amount of SCN2A that's there. And so on the left here, we have the state of a child with a loss of function, where you've got one normal copy and one copy which is just not making a protein, or sometimes is making a protein which doesn't do anything. And the hope would be with the ASO that you can increase the expression, and that then leads to something like the normal state with the equivalent of 2 genes expressing it. Now, work which Serena's probably going to show us offers some reassurance that even if you increase it too much, that doesn't seem to have very much effect. So the hope would be that this approach might have a slightly wider therapeutic window. It's worth pointing out that more expression is not the same as gain of function. But actually designing it in the first place might be more difficult. I think at the moment there are 3 concepts of how you might increase gene expression in SCN2A. So the first one is trying to skip what's called a poison exon. So there's the normal version of SCN2A, but there's also lots of extra bits which sometimes get included. Those are called exons. And some absolutely beautiful work done by Stoke Therapeutics did an experiment to try and to see what the potential was to remove these poison exons and increase the expression. And they found that SCN1A was a really good candidate for this, and have perceived that as a direction. So that skipping a poison exon is one possibility, so long as there's a poison exon there to be skipped. Another idea is to try and stabilize the transcript. This is a concept I've only seen mentioned in the last sort of year and a half. It's still, to me, it seems like a very emerging idea. It's not exactly clear how you design this so much as just try lots of different things. But that's an emerging idea which is potentially going to work well. And then on the far right, we have an idea that you can find another gene that regulates your gene of interest and decrease that one. And so I said earlier that there was an ASO which decreased the gene UBE3A antisense. What I didn't mention is that that antisense stops the production of a gene called UBE3A, which causes Angelman. And so that's the treatment in Angelman, which is looking like it's having a fairly dramatic effect in children. What it does is decrease a regulatory transcript, leading to an increase in UBE3A, and that's the therapy which you're trying to achieve. I think, so in terms of applying this to SCN2A, it's just going to show a little bit more here about the one which I think is the one we're pursuing as the first part of this. And this is the idea of a poison exon, which the example here comes from Dravet syndrome. And here's the data which has been published on their work in mice. And this is a thing called a survival curve. It's quite a simple concept. What you're seeing on the bottom axis is time. So the mouse is born at day 0. It's then given, some of them are given an injection at day 2, P2. And then you simply ask the question, well, how long do they live? And the percentage of the mice that live is shown on the vertical axis. So if we take the red one, this is the mice with the SCN1A mutation. And we can see that over time, it has a much more severe effect in mice than it does in human. Over time, survival drops really quite quickly. Around 30 days, we see a dramatic decrease in the number of mice that survive. In contrast, the mice without the SCN1A mutation, all of them survive. And the ASO given to the mice with the mutation almost completely reverts them back to normal from a survival point of view. So this is just, remarkable outcome. And this has led to 2 phase 1/2 clinical trials of this concept in humans. And so trying to find this starts off with seeing if there is a suitable poison exon to target. And that's work which is ongoing in my lab, also in collaboration with Nadav's lab too. And that was generously supported by a gift from some parents with a child with an SCN2A mutation. And we hope to have a sort of preliminary results in that really quite soon to see if there is something to be targeted there. And it's actually a genome-wide analysis. So we'll also be able to look at basically all the other causes of developmental delay at the same time. In terms of the other approaches of trying to stabilize transcripts and decrease regulatory transcripts, those will require a little bit more searching. This middle one's very new, and I'm still learning about how that might be applied to SCN2A. The one on the right, I have a couple of ideas about, but it's a bit more of a complicated screen than the poison exon. So we're starting there because it's the easiest one and because there's a very clear precedent of it working. And so that's a sort of an overview of the ASO side of it. I'd just like to say thank you to all the people I work with, and of course, particularly highlight Kevin Bender and Nadav and Serena, of course, we're going to see next. And then some of this work is being supported also by BioMarin. Thank you. I think we'll take questions afterwards, or do you want to take questions between me and Serena?
Does anybody have any questions yet? You can raise your hand or just unmute or put it in the chat.
I have one question. For an ASO for a gain of function, if it is too much, can it turn the child into more of a loss of function?
That's exactly the risk. And I think this is an emerging story, but the concern is that it can be quite hard to dose something, especially over a sort of 3-month cycle. And so if you give too little, nothing happens. If you give too much, exactly right, you might change the child into a loss of function, which of course can also cause seizures. And some work from Kevin's lab, which has just been published, just been on bioRxiv, anyway, somewhere, shows that actually the more you decrease SCN2A, the more you're prone to seizures at the other end of the spectrum. So I think that's exactly the concern, is that if you have the gain of function that leads to seizures, if you have loss of function that leads to seizures, so you've got to try and damp it down enough, but not too much. Now, who knows? It might be that we get lucky and that actually there is a wider therapeutic window in humans than we might expect. It might be that we learn new ways of regulating ASOs, which means that there's ways of adding on to it. But right now, that is exactly the concern. And what generally happens in clinical trials of these is that you start off at the lowest dose and then you increase it with each one, looking out for symptoms. And so I gave that example of Angelman, and that's actually a practical example of what this was like. At the lowest dose, they started to see some improvement particularly in social communication. Some of the children started talking for the first time, which is really dramatic. But as the doses increased, they started to notice a paralysis of the legs. And that was a thankfully reversible reaction at the injection site of the ASO. And so that's actually put the trial on pause for a year, lots of communication with the FDA, but now the trial's starting up again. And so when these go into humans for the first time, there's all these questions about dosage, which you've got to just pick a number and hope it's about right, usually informed by studies in primates and monkeys. And then you gradually increase it, hoping that you get more benefit than side effects.
Thank you. Anyone else have any questions right now, or should we move on to Serena?
One quick question for me.
Sure.
I know that ASOs have been used in a lot of neurological diseases from a bunch of different companies, but it seems like they do a great job of kind of getting into the periphery of the brain, but some of the deeper regions, they kind of struggle depending on the platform. Like when you look at SCN2A, what parts of the brain does an ASO ideally have to hit?
Yeah, that's a really great question. So I think my first reaction to that is in Angelman. So the best example we've seen of an ASO in the brain is, or in the central nervous system, is spinal muscular atrophy, where this ASO has a life-saving effect. But there, just like you say, you're trying to treat the spine. And you don't need to know much anatomy, that injecting into the spine is much closer to the spine than it is to the rest of the brain. But in Angelman syndrome, it's not the spine, it is the brain, little vague on exactly which bit of it, but probably, I think it's reasonable to think it might be similar to what we're trying to target in SCN2A. Right now, if I had to guess which bit that is, I would say that the frontal cortex seems like a leading candidate. The stratum as well, maybe. But that is an emerging story. In terms of cell type, I think we've got a slightly clearer idea. And the answer is we're trying to get into neurons, particularly excitatory neurons. But there are lots of unknowns here. So along with which bit of the brain can you get it to, we could also ask the question, well, what fraction of the neurons do you need to treat? And these are big unknown questions. So actually one of the other lines of research in my lab at the moment is using some novel 3D imaging to try and track where do ASOs get to, starting off in the mouse brain, but hoping to move that into larger model systems in the future. Exactly for the reason you say, because we need to know where these get to. But then there's other groups, like ASOs have become a very, very hot topic recently, I think in large part because of the experience in spinal muscular atrophy. And so there's other groups around the world working on improved delivery, the idea of giving it into the blood rather into the spine, trying to think about whether there's chemical modifications which target neurons more than other ones. And so there's a lot of different groups and parallel methods trying to solve these problems.
Got it. Thank you. One other follow-up question. You mentioned a few kind of knockdown approaches, but one that's recently been validated is using CRISPR to kind of also break the gene. When I think about that, you're permanently breaking a gene. In the context of SCN2A, getting like 30 to 40% breakage, which leads to like a net 30 to 40% knockdown. Like it's going to be cell selective, but like how would that work with like the networks of neurons, and like would that strategy potentially be effective?
Yeah, that's a good question. I think probably Serena might be touching on that a little bit on the CRISPR side of it. I would. So I characterized ASOs as being sort of gene therapy light. I think ASOs represent a nice stepping stone to answer those questions in a non-permanent way. Because as you say, when you make a permanent change, you want to be very, very sure you're making the right change. Serena and Nadav, do you want to comment on that side a bit more now or after your talk?
I think maybe after the talk.
Muting myself. Yeah, sure. Agreed.
That seems like a good intro to you, Serena. Why don't we go to yours? We've got a more complete picture.
Yes, thank you.
Pick up with the questions afterwards.
Thank you so much, Stefan. I guess I'll dive right in. I've already shared my screen. Does it look visible? Great.
Yeah.
Well, Stefan, that was an amazing talk. Thank you so much. And also a wonderful primer that leads directly into my thesis work. Just like to introduce myself quickly in case we haven't had a chance to meet yet. My name is Serena Tamura. I'm a PhD candidate at UCSF in Nadav Ahitub's lab. And today I'll be giving a brief presentation on my thesis work, where I'm developing the use of CRISPR activation as a gene therapy to rescue SCN2A haploinsufficiency in autism spectrum disorder. So in our lab, we're really interested in studying and developing treatments for human diseases caused by haploinsufficiency, which is a really big word, which means when one functioning copy of a gene is just not enough. Typically, we have 2 copies of each gene, one from each of our biological parents, and this gives us a full tank of gene levels. But in the case of haploinsufficiency, one of those 2 copies is mutated and rendered non-functional. So we're only left with 50% gene levels, and this can lead to a variety of human diseases. There are over 600 genes that are known to cause human disease as a as a result of haploinsufficiency, but whole exome sequencing predicts that actually more than 3,000 genes are haploinsufficient. And perhaps most relevant to my project and our talk today is that 89 out of the 102 autism-associated risk genes are known to be haploinsufficient. So we've applied the use of CRISPR activation as a therapeutic to transcriptionally modify and correct those gene levels in haploinsufficiency. You might be familiar with traditional CRISPR, which uses a guide RNA as a homing device and then directs these customizable genetic scalpels called Cas9 to cut out specific parts of the genome. CRISPR activation, however, does not cut the genome. Instead, it uses a scissor-free Cas9 called a dead Cas9. We call it dCas9 for short, and it binds to the genome. You can think of CRISPR activation as a delivery truck, and that truck uses the genetic coordinates specified by the guide RNA to learn where it needs to park itself on. I've loaded up my delivery truck with protein factors that activates the gene to make it work twice as hard. And in this way, we can rescue haploinsufficient diseases by rebalancing total gene levels. So my main question is this: if I can customize this CRISPR activation system to target the one healthy copy of SCN2A to work twice as hard, can I A, bring gene levels back to a full tank, and B, can I rescue the associated SCN2A haploinsufficient phenotypes? But first, I'd love to give you an introduction of our first proof of concept story. Navneet Matharu, an extremely talented scientist in our lab, led this study, which we published in Science a few years ago, where we used CRISPR activation to rescue SIM1 haploinsufficiency, which is known to cause a severe obesity phenotype. So what we did was we injected AAV particles that contain the CRISPR activator complex, that's the delivery truck fused with the transcriptional activator, and then designed guide RNAs to target the promoter of the healthy copy of the gene. We injected the SIM1 heterozygous mice at around 4 weeks of age with the idea that perhaps we'll be able to rescue that obesity phenotype. 9 months following injection, we were really excited to see that the CRISPR-A treated SIM1 heterozygous mice, shown by this yellow bar graph here, developed a body weight that was equivalent to the wild type mice, shown in tan here. Whereas our uninjected SIM1 heterozygous mice in orange and our no guide RNA negative control injected hets in blue here did indeed develop a body weight consistent with severe obesity. So these results really encouraged us to further explore CRISPR activation as a potential therapy for haploinsufficiency and to target more complex disorders like autism spectrum disorder or ASD. Stefan touched on this lightly, but the development of CRISPR-A, in my opinion, is especially relevant in terms of a therapeutic approach for targeting larger genes. Traditional gene therapy approaches packages a healthy copy of the gene's cDNA and then just directs it to the compromised tissue. But AAVs, which are the gold standard delivery mechanism, actually has a really modest load or size capacity. So not all genes are compatible with this method. In fact, if you see this dotted line here, all the genes to the right of this dotted line are too big to fit inside of an AAV vector and are therefore excluded from traditional gene replacement therapy. And when we looked at those genes that are too big, we saw that 44 out of the 102 autism sequencing consortium genes are too large to fit inside of an AAV. So as proof of principle for rescuing a gene that is relevant to autism spectrum disorder and also too large for traditional gene therapy, we decided to target SCN2A. We chose SCN2A because it's highly associated with ASD and it consistently comes up out of a top hit out of all the candidate ASD risk genes. We also understand its function, which is extremely helpful when studying a complex behaviorally defined condition like ASD. SCN2A encodes for that neuronal voltage-gated sodium channel NAV 1.2. It's expressed throughout the brain, but primarily in the cortex in excitatory neurons at this part of the cell that's called the axon initial segment, which is the part of the neuron that initiates and propagates the flow of action potentials. And that's the signal that our neurons use to communicate. Mutations in SCN2A are associated with distinct disorders in the clinical population. Loss of function mutations are associated with ASD and intellectual disability, whereas gain of function mutations are associated with seizures and epilepsy disorders. We're focusing in on those loss of function mutations because they fit the model of haploinsufficiency and are therefore a great candidate for CRISPR-A treatment. By using SCN2A heterozygous mice, we can study the physiological effects of carrying a loss of function mutation. And what we see here is that neurons from fully developed heterozygous mice, shown in blue, are firing action potentials that are 30% slower than wild-type mice, shown in black here. And again, these are age-matched neurons. And as a result of these slower action potentials, excitatory synapses are weaker in SCN2A heterozygous mice. The heterozygous mice show immature dendritic spine morphology, which are filopodial and long like spaghetti, as opposed to the wild-type litter mates, where you see the more canonical mushroom shape of the dendritic spine. Another quantifiable consequence of the speed deficit is the type of glutamatergic receptors that are found at these synaptic spines. And the filopodial spaghetti spines largely contain NMDA receptors and not AMPA receptors. And this confers a silent or an immature synapse. And we can take the ratio of the amount of current that flows through each type of receptor, which we call the AMPA to NMDA ratio, and we use that as a quantitative measure for the maturity of the spine. So I set out to rescue SCN2A haploinsufficiency with CRISPR-A using the same approach that we took to rescue SIM1 obesity. By shuttling my delivery truck DCAS9 loaded up with the transcriptional activator VP64, we send it over to the promoter of the healthy copy of SCN2A. And the idea is to upregulate translation of 1.2 channels off of that wild type allele and thereby overcome the ASD-associated cellular phenotypes. I started by designing 9 guide RNAs that target the promoter of SCN2A, and then I screened them in a mouse neuroblastoma 2A cell line, which is a common mouse cell line. I found that 3 guides upregulated mRNA expression by around 3-fold, so I produced those into AAV particles. I rescreened those 3 AAVs again in those Neuro2A mouse cell lines, and I found 1 guide upregulated expression by around 2-fold. So I selected that guide for the duration of my mouse study. Next, I wanted to see if the associated cellular deficits in SCN2A heterozygous mice can be rescued with CRISPR-A in vivo. So this is my general surgical pipeline. I stereotactically inject the medial prefrontal cortex of our heterozygous mice with our AAVs, which encode the DCAS9 VP64, and then the guide RNA. I inject unilaterally into one hemisphere so I can use the opposite uninjected hemisphere as an internal control. After about 4 weeks of incubation, I dissect out the prefrontal cortex of both injected and uninjected hemispheres, and then conduct qPCR, while the Bender lab, who we collaborate heavily with at UCSF, follows up on all the electrophysiological properties. We found that injected adult SCN2A heterozygous mice show a 1.6-fold upregulation in SCN2A mRNA expression when compared to their respective uninjected hemispheres. We were really excited to see that SCN2A CRISPRa not only upregulates mRNA expression, but it's also able to improve that action potential speed deficit. Here we have plots of the peak action potential velocities from individual cells. And you can see that the neurons isolated from the heterozygous mice shown in blue are firing 30% slower than the wild type cells shown in black. And we were really encouraged to see that our CRISPR-A treated heterozygous cells shown in green are firing action potentials just shy of that wild type velocity. Moreover, when looking at the function of the excitatory synaptic spines, we saw a rescue in the AMPA to NMDA ratio, again, which is our quantifiable marker for the maturity of the spine. And we saw that our treated cells have an AMPA to NMDA ratio equivalent to that of fully mature wild type neurons. One concern that we had in using CRISPR-A as a therapeutic to treating loss of function mutations is that we might inadvertently overexpress the gene and introduce a gain of function mutation, which again, we all know are associated with epilepsies and seizures. So in order to examine this, I injected wild type mice with our CRISPR-A constructs. I saw an upregulation of about 1.7-fold, again, compared to its uninjected hemispheres. And although we saw an upregulation of mRNA, the treated wild type mice continued to fire action potentials within the wild type range. So we saw no associated phenotypic change consistent with overexpression. And a working hypothesis behind this is that there's an upper limit to the number of 1.2 channels that can be expressed at the cell's membrane at any given time. And this might be an advantage of upregulating the gene off of its endogenous promoter. So to summarize my mouse work thus far, I've discussed how CRISPR-A can not only upregulate SCN2A mRNA expression in both wild type and heterozygous mice, but CRISPR-A is also able to improve that action potential speed deficit. And as a result, CRISPR-A rescues synaptic deficits and restores stability like the AMPA to NMDA ratio. And finally, we don't see any overexpression phenotypes consistent with neuronal hyperexcitability in CRISPR-A treated wild type mice. So in the last couple of minutes, I think I have a couple minutes left, I'd like to shift gears a little bit and share with you some preliminary work I've been doing to translate our mouse work into a more clinically relevant model. So we received 3 human-derived stem cell lines from the PAN lab at the Broad Institute. They previously published on these cells where they differentiated the stem cells into excitatory neurons. They first confirmed that the SCN2A heterozygous lines, of which there are 2, have about 50% mRNA expression compared to the wild type line. So they just did that for confirmation. And then when they looked at the functional characteristics of the cells, they found that the spike rates from the 2 SCN2A heterozygous cells are significantly reduced compared to the wild type's differentiated cell line. So I've been working on designing human CRISPR-A constructs and taking these stem cells and differentiating them into excitatory neurons in our lab to test whether SCN2A CRISPR-A can upregulate and ameliorate phenotypes associated with SCN2A haploinsufficiency in a human model. So this is the pipeline that I'm using to differentiate this human stem cells into human excitatory neurons. It's actually quite an involved process with many steps, and it takes over 3 months to grow these stem cells into excitatory neurons. But after about a year of optimizing this protocol during our COVID lockdown, I finally managed to make some headway that I'm excited to share with you today. Not only was I able to differentiate the stem cells into the correct cell type, but I was really, really happy to see this red fluorescent marker, which means that my human CRISPR-A constructs can efficiently infect these human excitatory neurons. But of course, next we wanted to see the effects of my CRISPR-A treatment. This work was done with Andrew Nelson, who's an incredible postdoc in the Bender lab. First, we took baseline recordings of the wild type and heterozygous differentiated neurons that I grew. And you can see just a marked difference between the wild type tracing shown in black compared to the heterozygous cells shown in blue here. And I was thrilled to see that my CRISPR-A treated heterozygous cells show a partial rescue, not only in their firing rate tracings, but also in their peak action potential velocities. So this is an exciting translation of what we see in our mouse models into a more clinically relevant model. And I think this preliminary data warrants further investigation. And I'm really excited to see what the future holds and all the lessons we can learn from this type of model. So I've gone over time a little bit, so I'd just like to quickly thank my collaborators on the UCSF SCN2A team, specifically Kevin and Stefan, Perry, and Andrew. Of course, my fearless leader, Nadav Ahituv. And I'd like to thank my generous funding sources, which include the Family SCN2A Foundation. And I'm happy to take any questions. Thank you so much.
Thank you so much, Serena. That was great. Do we have any
so
questions?
Sorry, my audio is not.
We can hear you.
You're coming through fine, Serena.
You're fine. Any questions from the group? Just raise your hand or just unmute yourself and ask. Or you can write it in the chat.
Serena,
Hi.
I'm going to jump in and ask a quick question.
I'm so sorry. Did you hear my presentation? I'm so sorry.
Serena, you're coming across. You're coming across
Oh my gosh.
It was impeccable as always.
No problem.
Oh my gosh. I just realized I tried to hear. I was like, oh, they can't even hear me.
It's all good. It's all good.
Okay. I'm so sorry.
Serena, on your last slide, it looked to me like your that middle column, it
Yes.
looked a little bit like the sort of hypo-excitability phenotype more than the sort of peak velocity. And so I'm wondering, are the neurons you're looking at, I presume, are relatively immature. So when you're maybe not seeing that complete peak rescue, might simply be that there's just such young neurons, you're not quite at the stage where you might see it.
That's exactly the case, Stefan. You know, in cell culture, these neurons are extremely old, but when you translate that to like real organisms, these are completely immature neurons. They probably correlate more to the first week in a mouse. So that's something that we're really aware of when we're doing this analysis, is that these are very, very immature neurons, but it still offers some sort of information into the human model, which is really exciting. But yes, they're very, very early, Stefan, and I'll show you at my thesis committee meeting next week more information about that.
Yeah.
That's great. Sean, do you want to ask your question?
Sure. Hi, Serena. Really exciting stuff there. Just kind of curious of the selection of AAV as a delivery. What AAV are you thinking about? You know, as you kind of consider, you know, clinical studies. I know AAV9 has been used with Zolgensma, but it also seems like, you know, that's a one and done solution. Some of the other kind of more new age CRISPRs are using, you know, APO delivery nanovesicles. It'd be cool just to hear your thoughts on all that.
Absolutely. That's a wonderful question, Sean, and something that we think about all the time. In the mouse work that I showed you, the AAV serotype is the DJ serotype, which is really commonly used in research purposes. We're exploring other serotypes that offer different ways to deliver the CRISPR-A treatment, including the PHP-EB serotype. I didn't show that today, but I've been piloting some intravenous delivery of my CRISPR-A particles using that serotype to give whole brain expression rather than a discrete direct injection. But that being said, Sean, you know, one thing that we think about is we're really focused more on what's inside of the delivery particles, right? There's incredible labs that are doing amazing work with epitope tracing to reduce the immunogenicity of AAV particles. There are other incredible groups working on those lipid nanoparticles. So I think that what we're showing here is that the general hypothesis of using CRISPR-A is viable. and can only be stronger with all these other newer and improved delivery particles. So maybe in the future, we'll synergize the two together.
Fantastic, thank you.
Thank you, Sean. Let me ask one question from the chat and then we'll move to Eloise. What variant types will this work in?
For CRISPR-A treatment as it stands right now, we're looking at the loss of function variants. That's really the classic model that this would fit directly with. But of course, one thing we want to think about is in the gain of function population where one allele has a gain of function mutation, perhaps if we were able to find a way to decrease expression from that allele, you'd still need to upregulate expression from the wild type healthy copy. So I think that this approach is still relevant to both sides of the mutation coin, in my opinion. I don't know if Stefan and Nadav want to add more details to that.
I think I would add the distinction. So within the loss of function, there's two groups. You've got the protein truncating variant. So that might be called a stop gain or a nonsense or a frame shift or a canonical splice site, something of that sort. In those, CRISPR-A seems a very, very clear straight shot at being the right thing to do. There's another group of loss of function with a missense variant. There it's a little bit less clear because what's going to happen is that both copies get upregulated. Now, if the missense leads to that one copy being degraded, then it'll work. If it leads to a protein which sits in the membrane but is just not functional, maybe a bit less clear. It might do, it might not, but it's hard to predict. And so I think, Serena, your comment about, I think a future complexity to this is going to be both with ASOs and with CRISPR, not targeting both copies, but being clever and based on an individual's unique DNA, targeting one copy. In which case, exactly as Serena says, on the gain of function, you can imagine diluting out the gain of function. Like if there's only so many slots, you increase the normal copy, and then instead of being 50% gain of function, you're 20%, 30%. Yeah. Eventually, there's going to be a number where maybe you just dilute it out. And so that's going to be another area for exploration. I think that does bring the slight challenge that to actually get one of these into the clinic is going to cost millions of dollars. And so it would be nice to have one therapy which works for as many people as possible, making it easier to get someone to spend that money to do it. If you're getting down to the stage where it's one therapy for one person, then you're in a very, very, very different place. Now, there might be somewhere in between there where if we were really clever at looking at common variants, which were near SCN2A, you might be able to get a sort of, there's 4 different therapies, you've got to pick the right one which fits with your DNA, but you might be able to that way upregulate the right one. But we need to show that the concept works before going down that sort of more complicated side of things.
Great, thank you. Eloise, did you want to ask your question?
Hi, it's Eloise's husband. Thank you very much for your work.
Sorry about that.
Serena. Just would love an understanding of, a realistic, but or maybe optimistic scenario. If you continue to see results that you're happy with, how long before this can be tried in humans?
Oh, that's a wonderful question, and something that I'll probably ask my fearless leader Nadav to help weigh in on, just because that's a little outside of my my scope.
Yeah, that's a great question. So there are a few companies that are already using this to develop therapies. There's one that's pretty advanced on SCN2A for Dravet syndrome that is hoping to do clinical trials probably in a year from now. So that will probably be the first therapy using this technology. And, if successful, then I think it's easy to basically copycat off that for other genes like SCN2A. So we'll see, and keeping my fingers crossed to see the results.
Thank you.
That's great, thank you. All right.
I would add to that so far, I think it's fair to say that we've had a fairly smooth line in the development of this. Is that fair? Like we haven't hit any fundamental hurdles yet, which is really very encouraging.
Yes.
That's great.
And there's been a lot of places where it could have been like, you give too much and you get seizures, and so I think if things continue like that, then we may be quite optimistic.
That's great.
Yeah, I would add that, my concerns are less the approach, but more the delivery and what's being delivered. There's not too much work yet on CRISPR and how immunogenic it could be. And there are other ways where you could use this. It doesn't have to be CRISPR. It could be zinc fingers or talons. Zinc fingers tend to be, less immunogenic. And also the activator itself, we don't know how immunogenic that would be, and then the delivery with the viruses, as Serena mentioned, there are great companies and people doing that. So that would be more my worry than the actual approach itself, which we've done, and now a lot of others on various other diseases and genes, and all of them show success. So I think it's more the actually how you deliver it and not to be immunogenic. That is the key now.
So from a very selfish point of view, it'd be great if SCN2A was the second disorder treated this way.
Right. See how it goes with somebody else first. Right. There was a question that I think you answered earlier, but would the changes be permanent with CRISPRa? Or would you need more than one treatment?
Sure. I think right now, just from my mouse work, this data was all based off of a single injection. And that's the advantage of this approach in targeting cells that are post-mitotic, meaning they're done dividing. So that means, which includes neurons or brain cells. So they should have stable expression of our CRISPR-A components in those cells for a long time to come. And I'm thinking about the proof of concept model, the SIM1 haploinsufficient mice that we treated, that lasted throughout their lifetime. I want to I don't know if I made this point very clearly, but the CRISPR-A treatment, I think one of the great advantages is that, again, it is not permanently editing the DNA. We're not actually cutting up the genome. We're not inserting it. We're instead sending in that delivery truck to just park itself and be happy sitting there for a long time. So I think on the flip side of that is that if we ever needed to, unpark itself or move off, that could be built into the system in the future.
We're also working on a sort of fail-safe method, or something you could give to try and stop it, which you just explore, thinking of if a problem did emerge, what could you do?
Right. That's amazing. Sean, did you have another question, or just have your hand still up?
Nope, I didn't.
Okay.
Sorry.
That's okay.
One point to add on that permanent question. We've got to remember CRISPR is quite new. Like it's less than a sort of decade of experience. And with CRISPR-A, it's even more new and more like sort of 3 or 4 years. When you ask questions about is it permanent, we just, there's no way of predicting, is permanent 10 years, is permanent 30 years, is it a lifetime? And that's going to be an emerging story once this starts to get translated into humans, because mice just don't live long enough.
Just as opposed to an ASO where you would have to keep injecting, or a small molecule, you'd have to keep taking something. It's more long-term.
Exactly. And having the ASO, we've seen in spinal muscular atrophy, there's quite a lot of people who start on the ASO and then you have the one dose, because there's no reason for the two not to be given together.
Okay. Leah, do you have a question?
Sure. So this is amazing work. Thank you all so much for explaining it so nicely to us. What kind of symptoms would you expect us to see a change in humans? Like, have you done behavioral testing in mice after they were rescued? And is there any change?
That's a great question, Leah. And we are not focusing on a mouse behavior in this study. I would say one thing that I'd like to see in the future is if this does go into non-human primate models, that would be a better model to understand the global differences in behavior. But our reliance on this mechanism of SCN2A haploinsufficiency, and we see this beautiful work in quantitative measures of the cellular phenotypes. My understanding is that it'll translate well to global behavioral differences that we just can't see in the mice in this model.
I think there's a long history of being worried about using mouse behavior as your endpoint. So I think best described in Fragile X, where there was a small molecule which created an amazing cognitive rescue in the Fragile X mouse, given to humans, did nothing, absolutely nothing at all. And one of the fathers quipped that it was, it's a great time to be a Fragile X mouse. So that the question about, if we were looking at seizures, the mouse is a pretty good model, not perfect, but pretty good. For social communication, for cognitive improvement, even for motor delay, it's really unclear what you can believe. And this is a problem across the whole of neuropsychiatry, the whole of developmental delay. We just don't know when you can believe the mouse. And then added to that, we do see a behavioral phenotype in the mouse, but you need to look at about 50-ish cases. And that's just, that's not practical. We can't be, your pace of discovery, if you're relying on something which needs 50 mice, is going to be just so slow. It's just not really worth pursuing. Kevin's actually got some interesting ideas of looking for kind of intermediary phenotypes in the mouse, which are maybe a little bit more quantitative and easy to measure. It's a really good idea, but it sort of remains to be seen how well it's going to work. But as Serena put it, we have a molecular mechanism which is quantitative, which sort of captures something. So when she's showing AMPA to NMDA ratios, if I gave that the name learning, that's kind of what you're seeing there. And so it seems like a fairly robust molecular marker. But until we put it into a primate or even better, a human, there's really no guarantee of the rescue. From the Angelman experience, what they saw there was that the biggest improvement, I believe, was in the social communication and language domains. But the therapeutic window, duration was fairly short. And what I would expect, and here I'm speculating wildly, but if normal development goes like this, and in a developmental delay that everything is delayed, I think the name is quite good. You probably can't make up for lost, but what you can do is get back to the curve you should be on. And so it's unlikely to ever be a cure, but you might be able to accelerate the pace of development.
So is it fair to say that they might have an ability to learn? They're not going to all of a sudden be singing and skipping down the driveway, but they might have the ability to learn new skills once corrected.
But that would fit with what's being seen in Angelman. And bear in mind, that's based on, I think, 5 individuals. This is all this question about what is reversible is one of the biggest unknowns in the field. And I honestly cannot see any way of answering it apart from in humans.
Right. I got another question in the chat. Would it matter what age this treatment is given?
Yeah, that goes directly into what Stefan was just talking about. But the age of mice that I can speak from the mouse model perspective, this was all in pretty developed, mature mice, which we were thrilled to see any sort of cellular difference, because we didn't know that we could sort of kick into gear this rewiring of the brain at such a late point in development. So that offered a lot of confidence and excitement for us in terms of our research in a preclinical sense.
It's encouraging.
Yeah, absolutely.
Yeah, great.
Simply earlier will be better, but whether there's any plateau to that. I always have the model in my mind is social communication like an eye or an ear. So if you cover an eye from birth and then at the age of 4 uncover it, that eye will never work, because there's a critical window, and the loss of that connection is, once it's not made, it never gets made. In contrast, if you can't hear, at a later age, you can put in a cochlear implant and you can hear. And so there seems to be less of a critical window and more of an ability to reverse it later on. And so the question is going to be, for something like learning or development or social communication, which of those two paradigms is more accurate? The early angel and mouse work suggested it was more like an eye. And so they were showing there that if you didn't do it prenatally in the mouse, you just never rescued it. But then we see this in the humans, and the answer seems to be more like an ear. And actually there is an ability to rescue it later. And so I think, again, it's going to come down to the experience in humans. That's going to be one thing we need to measure very, very closely. And if ever we get to the FDA, we're going to need to be asking to treat as early as possible. And interestingly, in Angelman, it was the FDA who drove that decision to go for the younger age group. So I think they are very aware of that need.
Interesting.
Partly because you also might need to build the you might see a bigger change in younger children, allowing you to give a bigger dose in older children to try and affect more of an improvement.
Mm-hmm. Any other questions out there in the group? We still have plenty of time.
Got one more, Jenny.
Yeah, go ahead, John.
A lot of our kiddos have symptoms, outside of seizures, like GI issues, sometimes kidney issues, that seem to be some genotype-phenotype correlation. As you think about a gene therapy, would correcting it in the brain resolve many of those, or would you need more systemic administration?
So that's a really good question. Simply, I don't know. I can offer you a story of hope, which seems appropriate to this group, but also a sort of pessimistic one. So the hope would be, I recently saw a paper looking at the microbiome in autism, and you get the microbiome is sort of the germs, the bacteria in your guts, and they're really kind of important to how your guts work. And if you look at individuals with autism, you see quite a different microbiome to children without. And so there's this idea that that's sort of the cause of the GI problem. But there was some really nice work done by a group in Australia where they did a really nice experiment, did it big enough, and showed that actually it's kind of back to front, that the microbiome is downstream of the autism diagnosis. And essentially, if you have a child who's got restricted interests, who is a picky eater, who is difficult to communicate with, that dietary change is probably what's changing the microbiome. And so that might lead to a theory where it is the brain changes are leading to behavioral differences, which partly contribute towards the GI symptoms. And so in that case, if that was the case, you might expect there to be some improvement there. The flip side is that people always describe the gut as being like the third nervous system. So it's the biggest collection of neurons outside of the central nervous system is in the gut. SCN2A may play a role there. We don't see a lot of evidence for it in the gene data, but it may do, in which case you might need to sort of try and treat it there as well as in the brain. Now, actually, the ASO probably won't get there unless it's given systemically, whereas the CRISPRa might do. The virus can get out of the brain. It would depend a bit on which virus you pick. So I think that's just a question we're going to have to see. The kidney's a harder one. And with the kidney, you've also got all the nasty medications which are being given, which are probably having an impact on the kidney too.
Thank you.
Thank you. Leah, do you want to talk about the post you just put up?
No, I was actually an accident. But this
Okay.
one is,
sorry I mentioned it.
no, I was looking up some of the articles that Serena had mentioned in her talk. So I just grabbed the link. But anyways, the link tree that I just put up was Carla would like to talk about that.
Floor is yours, Carla.
So the link tree is the foundation's link tree to a lot of direct access. So there is a link on here for all the past Hope series talks, and also this one will be uploaded there, so you can click on the link tree and other things about SCN2A and about families SCN2A.
Where do you find Linktree, Carla? You just go to Linktree.
Yes, you can find Linktree on our Instagram. It's on our bio right there. Also, if you type in Linktree and also Families SCN2A, you can find us there too as well.
Great. For us that are not as technologically apt.
And it's also in the chat. Leah put it on there in the
Yes,
chat.
that's great. Thank you. Any more questions out there? Anyone? Anyone?
Bueller.
Bueller?
I'll go ahead and close up, Jenny, if that's all right.
Yeah, go right ahead.
All right. Well, I mean, I don't really know what to say except for wow, it was an amazing session. And thank you so much, Stefan, Serena, and Nadav for volunteering your time to spend with us on a Sunday to help us understand this exciting work better. It gives us such hope for our kids' future. And thank you to the families who joined us today also. Your questions and comments were very insightful. If you're like me, that you'll probably think about this the rest of the day, maybe a few days, and you'll think of questions later as you digest the science. And just so you know, the volunteers and the staff at the Families SCN2A Foundation are here for you. You can send us those questions anytime, and we'll do our best to get them answered for you. There are 8 more of these talks in the RX Hope virtual series, so be sure to register for them in advance. You can find the link in Linktree or on our website, and we will be making a recording and then we'll put them on the Linktree as soon as we can. There is still so much work to be done, and we could really use your help. As these researchers are doing their amazing work, we're fighting hard to be clinical trial ready. So there's 2 major ways to get involved right now. One is through our annual campaign that we just extended till the end of August. So you can check that out on our website. And also participation in the SCN2A clinical trial readiness study, also in Linktree. So I'm just going to leave you now with best wishes for a safe, quiet, calm, and seizure-free rest of your weekend. Thanks, everyone.
Thank you.